Okay, so thank you for giving me the opportunity to present some of the data I've been working on. So I've been working on the ATP synthase mainly throughout my postdoc work in Frankfurt, and I started to work also on it here in Berkeley. I've been here for 3 years. So ATP synthase is, to me, a very important machine. It's definitely the most coolest protein in biology. And it's essential for all life on Earth to generate energy that we live in. So if we look around the world, around Earth, we have many different types of ecology. So we've got things like we've got oceans, we've got deserts, we've got Arctic tundra, we've got the deep sea fence, and also the sulfur lakes, thermophilic lakes in like Yellowstone Park. And wherever we look at, we always have organisms. So no matter what sample we take from the environment, there is always something living there. But the environment they're living in is very different. Now, what unites all these organisms is a desire to make energy. Without energy, they can't live. And the molecule which they're all trying to make is called adenose triphosphate. And that's found in the center here. It's a 500 Dalton molecule, chemical, and why it's so important to life is that it's got that last bond, which is highlighted in yellow there. It contains a lot of energy. And if you break it, you're releasing 34 kilojoules per mole of energy into the environment. And if you can capture that, you can actually use that energy to power other processes in the cell. For example, we like to move. So for our muscles to move, we need to generate energy in order to contract our muscles. And that is generated by breaking that phosphate bond at the end of that small molecule. So in order to generate that molecule, to have that molecule ready to break so that we can power any reaction in our body, we need to make it. Now, all of the organisms in any ecosystems are all trying to make this molecule. And it's generated by metabolic processes. Every single environment has different types of metabolism that it uses to generate this molecule. And what it's using depends on the environment it's in. So we are organoheterotrophs, so we need to eat food to get our energy. So we get our carbon supply, we get our reducing equivalents from the food we eat. So we rely on organic molecules. The other types of organisms we have are like plants and cyanobacteria, and they use the energy from the light to generate this molecule called ATP. And these are shown on the second line down, which is lithoautotrophs. Okay? So they get all their energy, so their reducing equivalents, their carbon sources from the air. And we can actually categorize all the organisms in life into these 3 different types of metabolisms. Now, if we look a bit closer at these metabolic pathways, I've just got 3 shown up here. The first one on the top left, is oxidative phosphorylation. This occurs in the mitochondria in our cells, and that's what allows us to get the energy in the cell that we need to do for movement. The bottom left is the photosynthesis. These are the proteins involved in generating ATP in plants and cyanobacteria. And on the right-hand side, you've got the pathway which involved in generating ATP from methanogens. So they use methane as a source of getting energy. Now, if we look at them, we can actually divide those 3 pathways into 2 simple principles. First of all, what's indicated in red here is the proteins. These are the ones that take what they have in the surroundings. So we take food, we break it down. And what they're doing is they're generating a chemical gradient across a membrane. In the case of oxidative phosphorylation and photosynthesis, this is generating a proton gradient across the membrane. And on the right side, the methanogens are generating either a proton or sodium gradient across the membrane. So what we're trying to show here is that we've got many different organisms in the world, but when it comes down to making energy, what they have is a group of proteins which are dedicated to using the environment's resources around them to generate this electrochemical gradient across the membrane. Now, the second part of the metabolic pathway is shown here in blue, and that's using the energy stored in an electrochemical gradient to drive the synthesis of this ATP molecule, this molecule that's going to give you the energy of life. And that's what we're going to be talking about today, is no matter how diverse the ecosystems are, what organisms you're looking at, they all have the same principle. They all have this ATP synthase molecule, which takes an electrochemical gradient. They use the energy in that gradient to generate ATP. So we've already seen, just put this slide up to make sure that we're all familiar about the kingdom of life. So we've got three main lineages. We've got the bacteria, we've got the archaea, and that we've got the eukaryotes. And there's two endocytosis events where bacteria were taken up by the archaea, which generated mitochondria, which we have in our bodies, which generate energy, and chloroplasts, or the plastids, which is what plants use to capture the light energy. Okay? So if we look at the ATP synthases that we find in all these organisms, these are the ones we see. So these are responsible for generating ATP. There are three different varieties. We've got the F-type, which we find in our body and in bacteria. We've got the A-type, which is found in archaea. And we've got another type, which is called phi-type, which is very similar to the A-type, which is found in the vacuoles. Now, the F and the A-type generate ATP. So what they use is the energy and electrochemical gradient of protons to drive the synthesis of ATP from the breakdown products of ADP and inorganic phosphate. And the phi-type is very similar, but it works in reverse in that it hydrolyzes ATP to make ADP. And in doing this, it pumps protons into the vacuole, which acidifies it, which breaks down all the nasty stuff in the cells we never want anymore. But when we look at that, you can see there's a similarity in these protein structures. They all have the globular head domain, which is called the catalytic domain, and this is responsible for putting that phosphate, the third phosphate, on an ADP molecule to make ATP. We've also got a rotor area, which rotates in the membrane, which is the driving force of driving the catalysis in the catalytic domain. And we have a stator, which holds everything stationary to the membrane. So it allows the rotor to rotate without rotating the catalytic domain. Nature never makes anything fresh. So it is thought that the ATP synthase is it's come from an RNA helicase, which is involved in the translation of RNA into protein, and a proton input channel. And that these are sort of held together by two stators, and this is how it evolved into the ATP synthase. Now, the reason it's thought about that is if you take the catalytic domain of the ATP synthase, it originates from a family called the AAA plus ATPase family. This family consists of identical subunits that form like a donut shape. So each one of the colors on the left-hand side is a different subunit. And then if we take out one of those subunits, the nucleotide, the ADP, sits into the little snugly into the cavity between the two domains. And on the third side, we've got a zoom-in of the active site. And so one subunit is sort of snuggling around the ATP molecule, and then there's a star next to an arginine residue. That's really important, because that arginine residue comes from the neighboring subunit. And as there's conformational change in that hexamer, that arginine gets pushed into the active site, and that what causes the reaction to occur. And in this case, it's hydrolysis of the ATP. So this is what the cell uses to break apart that last phosphate bond. But in the ATP synthases, we've had a slight modification of this protein. So what we have is we don't have identical subunits anymore. We've got alternated subunits, alpha, beta, alpha, beta, alpha, beta. And there's only one catalytic site between the alpha and beta molecule, and the next one is non-catalytic. And the other modification that happens is that these 6 subunits work together. So if you look at the right-hand picture at the top, you can see there's different size gaps between the subunits. And this allows the ADP molecule to come in. Then there's a conformational change, which causes that gap to close, which pushes the arginine in. which does the catalysis, and then there's another conformational change which allows it to come out. So you basically got 3 different sites which have different stages in the production of ATP. What changes those conformations, what drives the conformation, is the rotation of the central stalk. So on the left-hand side, you've got the catalytic domain at the top. You've got a stator, which is holding the catalytic domain stationary relative to the membrane. And then the rest of it is sort of like a ring structure with a stator going up into the middle of the catalytic domain. And as the rotor rotates in the membrane, it's rotating around the subunits in the catalytic domain, it's pushing against each one of them. And that causes those subunits to change shape. And that powers the reaction of putting that last phosphate group onto the ATP synthase. So in the membrane, what we have is we need to get the proton gradient through the membrane. And what we have is 2 half channels. And you've got a membrane potential across that membrane and a high concentration of protons on one side. So the protons go through a channel to the middle, goes onto the ring structure, and then the ring rotates around. And then it comes out on the other side. And this is the same in every single ATP synthase. So this is a movie showing how the structure of the membrane domain is going to turn around to the base. And if you go through it, you see on the left-hand side here a ring structure, and on next to it is a big alpha helix, which is in blue. So we've now got it in yellow and in blue. So what we have is that ring structure in yellow, and then we have these tilted helixes. And what these tilted helixes is doing is sort of holding the C-ring in place and also generating 2 hydrophobic half channels, which allows protons to move in on one side and then move out on the other side. And in the middle of that helix, there's this residue called arginine, which binds to the C-ring and causes a salt bridge, which prevents the protons going through straight away through the membrane from one side to the other. And it forces it to bind to the C-ring and causes it to rotate. So if we look here, we're going to look closely at this membrane domain. We're going to see in pink is the membrane. It gets pulled down to center. We're going to rotate it, or should now rotate, and you can see right into the center. And so that's the matrix half channel. So that's continuous with the matrix side. So any protons that reach that cavity come straight out into the matrix. And on the other side, there's a tiny little hole which allows the protons to go in to the membrane. But it can't go out the matrix channel because it's blocked by that arginine residue. So instead, it binds to an acidic residue in the middle of the C-ring. And when it's bound to that residue, the residue sort of bends into the C-ring and allows it to enter the membrane. So what's happening is on the left-hand side, you've got the C-ring in yellow. You've got the blue, which is the alpha helixes. And you've got this little pocket where the protons are coming in because it's been driven in there by a proton gradient and a membrane potential. It interacts with an acidic residue on the C-ring, which then folds into the structure, which makes it hydrophobic and allows it to enter the membrane. And then the ring rotates around and comes through to the matrix channel, which is aqueous, which allows the protonated acidic residue to open up, and the proton will go out because there's a low proton concentration in the matrix. And then that acidic residue will bind with the arginine, which is in the center, which pulls the C-ring around. So you get this rotation. And so and this is found now. This was shown a couple of years ago, and it's now found in bacteria, the phi-type ATPase, the A-type ATPs, and the F-type ATPases. They're all identical. This mechanism of getting the protons across the channel is the same in every single ATP synthase we study. What is different is the size of the C-ring. So the overall structure of the ATP synthase is identical, but the size of the C-ring changes, and also the residues on the entrance channel. And this allows those ATP synthases to just change slightly so that they can be more efficient in the different bioenergetic systems that they are found in. So in the mitochondria, we only have 8 and 10 C-rings, whereas in the plants, we have 14 subunits in the C-ring. This means the ratio between the amount of protons you need to generate ATP changes. So in the 8 C subringics, you need 8 protons to generate 3 ATPs, and in the plants, you need 14 protons to generate ATP. And this we think is because in the plant chloroplasts, the ATP synthase is driven more by a proton gradient, whereas in the mitochondria is driven more by a membrane potential. And there's a smaller difference between the concentration of protons on one side of the membrane to the other. So the other part of the structure is the stator. The stator is really important because it holds the catalytic domain stable relative to the membrane. So the rotation of the rotor is not lost when it goes into the catalytic domain. So holding the catalytic domain stable is really important so that you get that rotation of the central stalk in the middle of the catalytic domain to force the conformational changes that occur in the catalytic change, which is required for making ATP. Now, if you look at the general F-type ATPase, which is found in chloroplasts and bacteria, and then the feed type and the A-type, you see the status are very similar. It's just 2 alpha helices coming down, attaching the catalytic domain to the membrane. If we go to mitochondria now, the peripheral stalk is very different. And this is the part of the structure which has shown the most evolution and changes throughout the kingdom. And that's because in mitochondria, ATP synthase has a different function. It is actually important in the dimerization of the ATP synthases, which are important in shaping the crystalline membrane. And so this is showing you a little tomogram of, this is a slice through tomogram of a mitochondrion. You've got the outer membrane, then the inner membrane invaginates. And what's shown in the yellow arrows is 10 nanoparticles, which are actually the ATP synthases. We can look through the mitochondria, and you can see we can look at the membrane structures. So we're going back through it. We have segmented out the membrane in blue, and we positioned those 10 nanoparticles in yellow. You can see they form these rows, double rows along the highly curved edges of the crystalline membrane. We can now cut out subvolumes around those particles and average them together. And this tells us that when we average them together, we get this structure, which you can see is the ATP synthase, but it's dimeric. Okay, so this is only found in mitochondria. All the other ATP synthases in any other species or organelle is actually monomeric. We could take this average now and put it back in space and look at how it's formed in the membrane. And this is shown in this diagram on the right-hand side here. And we'll actually see that it forms rows along the outer edge. Okay, so it's actually involved in shaping the crystalline membrane. So though the ATP synthase is identical throughout archaea, bacteria, and chloroplasts, in the mitochondria they're different. This is also shown here in these different structures inside the mitochondria. So you can see the peripheral stalk has a wide variety of shapes, and the proteins in there are not evolutionarily conserved between the different species. But the part that's related to the functioning of the ATP synthase is conserved. And one of the reasons for this is the ATP synthase shape the cristae in mitochondria. Okay, so in conclusion, all life requires energy, and that energy comes in the form of this small molecule called ATP. To generate the ATP, you need 2 things. You need an electrochemical gradient, and you need the ATP synthase, which uses that electrochemical gradient. The electrochemical gradient can be made in different ways, but the way the ATP synthase is the same and is highly conserved across life. And the only variation we see is in that stator, and we only see that variation really when we go to mitochondria because the ATP synthase has got this second function. And so basically, the ATP synthase is a very essential machine to life, and it's conserved throughout all kingdoms, no matter what ecosystem it's found in. Okay, and then that's it. So.
question?
So you described how ATPase works across the domains, et cetera, et cetera.
Yeah.
Do we have any idea how it came to be, how it evolved chemically, prebiotically?
Yes, so it is thought to come by so the catalytic domain is similar to an RNA helicase, so these AAA plus ATPases. So it's thought that the AAA plus ATPase came first and translates the RNA. And then as the RNA can be transported through membranes, and it needs a transporter. And it's thought that the ATP synthase is a combination of this helicase with the proton transporter.
Okay, I might ask you for a reference on that later,
Yeah,
if there is one.
I have it written on my slide, so I can do that.
Because the creationists tend to use this protein as their argument.
10-minute break. Oh, we have one more question? Or?
One more question.
So some organisms get on without this, right? The ones that use glycolysis to make ATP. And could you say how that fits in?
So there's certain parasites have gone into our body. So like Trypanosome brucei. They've lost their ATP synthase because they use the ATP produced by the host. So they don't have ATP. There is some organisms, so in our cells, there is some cells which mainly use glucose if you've got a good supply of glucose. But the amount of ATP you produce with the glycolysis alone is very low. So for surviving in extreme environments where the food is very rare, they always be able to produce ATP through the oxidative phosphorylation or one of these metabolic processes. So I would say that the loss of is more of a loss of ATP synthase in some species than a gain of function in others. So it depends on what the availability of glucose is in the environment.
So are you
It has ATP synthase.
I got turned off.
Oh, thanks. Are you saying that the ATP synthase-based organisms with these with these wonderful motors evolved first, and then later on some organisms lost it and just use glycolysis or use a host's ATP? I thought that the earlier fermenting glycolysis types evolved first, but I could be totally wrong on that.
Yeah, you might be right. I haven't looked into it that much. But the ATP synthase is very conserved throughout all different organisms. But the proteins used for the metabolic pathways is highly variable. So like the methanogens use a different method to, say, plants, which use a different method to, say, us. And so glycolysis and the ones that just use glycolysis is a very small subset, and it's not so broadly applicable because there's only a subset of the metabolism that's available in life in general.